has been director of the CREA Serial Research Center in Foggia, in Italy, leading a group of about 25 researchers with a main interest in breeding, genetics, metabolomics, and processing technology of durum wheat. Since 2010, Dr. Cattivelli is the director of the CREA Research Center for Genomics and Bioinformatics, where he coordinates a group of about 40 researchers working in plant genomics, plant physiology, and bioinformatics. Dr. Cattivelli research activity has been focused on genetics and molecular biology of cereals, mainly barley, wheat, and rice, from plant breeding to gene cloning, QTL analysis, and genome sequencing for identifying and investigating key genes involved in the expression of agronomic traits. He has participated to the international initiatives for the sequencing of bred wheat and wild emmer genomes and coordinated the sequencing of the durum wheat genome. He is also the Italian representative in the Research Committee of Wheat Initiative, an international organization for the coordination of wheat research endorsed by the G20 agricultural ministers. And in the frame of Wheat Initiative, he is also co-chair of the expert working group on durum wheat genomics and breeding. He is also member of the editorial boards of many journals, Plant Science and Journal of Cereal Science, and many other journals. Scopus registers more than 200 publications and more than 10,000 citations for Dr. Cattivelli, which a H index of 50. I can keep going and talking about Dr. Cattivelli experience, but for the sake of time, I will stop here, and for sure, you can get information about his experience in the internet. Today, he is going to present a global platform for leveraging tetraploid wheat diversity. Before giving the floor to Dr. Cattivelli to start his talk, I would like to mention that the conference will take up to 40 minutes, and then we will have the discussion that will take up to 50 minutes. For the discussion, please post your written questions via the chat on Zoom. Please fill in your name and affiliation before the question to know you, and do not wait until the end of the conference to ask them. You can post your question in English, but also in French if you want. We look forward to listening to you, Dr. Cattivelli, and to knowing more about these exciting topics. The floor is yours, Dr. Cattivelli.
Thank you very much for this invitation and for this super introduction. So I will try to do my best to present you our work. And today we talk about biodiversity, so genetic diversity in durum wheat. So durum wheat is a tetraploid wheat that is different from the bred wheat. It is hexaploid, and is a main crop in the Mediterranean region, and this is why probably is a major topic for Italian research. The work I'm going to present you today has been made mainly by my research group together with the research group of Roberto Tuberosa from the University of Bologna, and in the frame of this wheat initiative activities. Roberto and myself, we coordinate the expert working group on durum wheat within the wheat initiative frame. And most of the people that are involved directly or indirectly in the work I'm going to present you today are part of this networking activity and this network of scientists working on durum genomics. Okay, so before start, I have only one introductory slide. Okay, and this introductory slide just to give you a few key steps in the durum wheat domestication and selection process. This will be very important to understand the next step of this presentation. So when we talk about durum wheat, you should know that the progenitor of the current durum wheat is called wild emmer. Wild emmer or Triticum dicoccoides. The white emmer is the progenitor of all tetraploid wheats, and there's a typical brittle rachis phenotype. We see here that the rachis of the spike is broken down when the seeds are ready, are mature, and so the seeds can be dispersed naturally. Then, about 10,000 years ago, in the Near Eastern fertile creations, a domesticated emmer wheat was selected from wild emmer via the selection of plant carrying mutation on the two genes conferring the non-brittle rachis phenotype. So this is the first transition from wild emmer to domesticated emmer. Then, after the domesticated emmer, durum wheat land races in Italy were evolved from cultivated emmer through a process of selection for free threshing and naked form. So durum wheat is a naked form, while emmer is a covered form. And the first durum wheat dates back about 6,000–7,000 years ago, but the durum became really established as a prominent crop only about 2,000 years ago, particularly in the south of Italy and the north of Africa. And then from the durum wheat land races, plant breeding has selected the cultivars first from land races and then applied genetic knowledge. So actually the elite cultivars derived from the original land races that were grown they were still very popular till say 100 years ago in the south of Italy and in all the Mediterranean country. And so there is a process going directly from wild emmer to modern elite varieties. This process has three major steps or three major transitions. The first from wild emmer to domesticated emmer, the second one from domesticated emmer to durum wheat, the third one from durum wheat to plant breeding. So this is what is generally known about the evolution and the domestication and evolution of durum wheat. Moving from this information, we try to get more in deep and to see how this process has an impact on the genome configuration of the plant. So our idea is to understand what makes a durum wheat cultivar, elite cultivar durum wheat, different at genetic level from the ancestors, including durum land races, domesticated emmer, and white emmer. To approach this problem, we organize a panel of biodiversity. We call this a sort of a global platform for biodiversity in tetraploid wheat. This global platform is made of two panels, basically. The first one is called Tetraploid Wheat Global Collection, and is a perfect tool for evolutionary studies. This tool, or this panel, TGC, is made of about 1,800 accessions and contains all tetraploid subspecies while they're exactly well represented from all different regions in the world. Then we have another panel which is more focused on breeding, and it's called Global Durum Panel. This is about 1,000 accessions and is made mainly on land races and modern varieties. And there are a number of accessions, about 300, that are in common with the two panels that allow to transfer the information, the result from one panel to the other. Okay, let's start to talk about the first, the TGC. So TGC is a panel that was developed together with the sequence of the durum wheat genome. It was published 2 years ago. And the idea of this panel is to make a collection, so-called top-down approach, because it's made of wild, cultivated emmer, land races, all subspecies of Triticum turgidum, and it's been made with the aim to represent all the genetic diversity existing in a gene bank for durum wheat, all the genetic diversity which are known for tetraploid wheat. So altogether, we believe that this represents the entire evolutionary history of the tetraploid wheat from ancestral, from progenitors to wild emmer to the modern varieties. The panel has been completely genotyped with the 90K SNP array from Illumina. And then, now this is the composition of the panel. See, the panel has some major components. So there's a lot of representation from wild emmer, a lot of domesticated emmer. Then we have a large representation from land races, about 800 accessions, coming from every region where the durum wheat land races were grown. And then we have also modern varieties, or elite varieties of durum wheat. And then there is also a representation of a lot of subspecies like Carpicum, Polonicum, Turanicum. They are probably not so frequent, not so popular, but they represent particularly niche where the cockum was, or the domesticated emmer was selected, and so they are adapted to specific region, probably in different regions of the world. Okay, the total panel is 1800, and using this panel, first of all, we can work on the population structure. I mean, the panel was completely genotyped, so we had about we use about 17,000 SMP, all of them anchored on the genome. And we approach the analysis of the structure of this population, this panel, using 3 different independent approaches. The first was this one here, where we make this phylogenetic neighbor journey trees. And what we find here is the final output. Clearly, there is already a differentiation of, in different clusters, and each cluster represents a different subgroup. For instance, we have in this position the wild emmer wheat, those 2 clusters. The wild emmer wheat is divided in 2 clusters according to the 2 regions of the fertile crescent. Then near to the wild emmer wheat, we have domesticated emmer wheat. So that makes sense because domesticated emmer wheat derived from is the first plant arriving from wild emmer. Then there are 2 clusters of domesticated emmer wheat and durum wheat races that come from Ethiopia. So Ethiopia region is completely apart compared to the rest of the evolution of durum wheat. And there are 2 clear clusters which has made only of plants coming from Ethiopia. Then the yellow cluster presents durum wheat land races, and you see there are many durum wheat land races, and very nearby the wheat land races, you find the cultivars, the modern cultivars durum wheat. So perfectly everything fits together. The idea I showed you at the beginning, from a wild emmer to domesticated emmer, then there's a selection of land races, and then of the cultivars. Then there are a few other small clusters representing different subspecies, which most of them are within the genetic diversity represented by the land races. Well, this is the first approach to understand the population structure. A second approach, we can use the same data set to make a principal component analysis. And this is what we show here. And again, you get a very similar take-home message. From one side, on the extreme left of the figures, there are the wild emmer wheat, the blue and the light blue symbols. From wild emmer wheat, there is a number of population of domesticated emmer, the one in green. Then we have durum wheat land races in yellow, and on the opposite, compared to the wild emmer, we have the durum wheat cultivars. Again, there is a clear representation. There are major groups. They're more or less the same major group represented in the previous analysis, and those major groups clearly separate one each other. There is you can imagine to have a flux of diversity from the with the wild and the domesticated in the modern cultivar, the 2 extremes of the genetic diversity universe. A third approach to see again the population structure of this panel is being run using this software called admixture, and again you get this very similar take-home message. So first of all, if you look at the K3, for instance, there is a separation between Mediterranean cultivars, Mediterranean land races, the one in yellow, from durum wheat modern cultivars, the segment in red. Again, we have the Ethiopian wild durum wheat land races, the one in green, which is different from the Mediterranean durum wheat land races. And on the left side of the structure, we found the wild emmer wheat, the blue, and the domesticated emmer wheat. So again, these all 3 analyses, although they work on it using different statistics, end up with very similar results. So we have a really subpopulation that are very homogeneous within the subpopulation, and that represent different step or region of the domestication and evolution of durum wheat. Okay, let's keep in mind these 3 major results and the overall population. We try to understand more about the impact of evolution and domestication on the genome of durum wheat. Well, can be okay. So, what is the philosophy of this work? First of all, we try to compare 4 different subpopulations. So the subpopulation are wild emmer wheat, domesticated emmer wheat, durum wheat land races, and durum wheat cultivars. we did not include in this analysis the accession from Ethiopia because they're quite apart compared to the rest of the analysis, and because these are really a very special niche of the evolution, so this was left out of this analysis. But the all other subpopulation have been considered together. And the first approach was to use the molecular data to look for the diversity in each locus within each population. And then what you find here, we compare the diversity of each subpopulation along the entire genome. And in this very nice graph, very colorful, you find that every circle here represents a different population. So the one on the top is a wild MR wheat, then a domesticated MR wheat, durum wheat landraces, and durum wheat cultivars. I understand that if you see like all together, this result might be difficult to understand, but I can zoom in and give you some example of the type of finding we had in our results. So this is a concern chromosome 6A, and what you have here is a 6A of wild MR wheat compared to the 6A of domesticated MR, durum wheat landraces, and durum wheat cultivars. This index represents the diversity, so the level of diversity in this region. And clearly you can find that the domestication, the first step, has reduced the diversity in this region. So we think we might speculate that the domestication here is make a selection of some region, reduce the diversity in a specific position of your genome. This is a very large region, more or less half of the chromosome 6A, but still is a very nice example how domestication can impact on the genomic structure of the genome. But there are other examples. For instance, here you find a situation where there is a reduction of diversity, but only in domesticated wheat landraces and in domesticated wheat cultivars, in durum wheat cultivars, and not during the first transition from wild MR to domesticated MR. Or you can also find another example in chromosome 2B, where a strong reduction in diversity has been detected only in modern cultivar. So overall, we can say that domestication and selection have reduced genetic diversity, but this loss of diversity is not homogeneous along the genome. There are specific regions where this loss of diversity is much stronger compared to the other regions. Okay, so you might use this information as an indication of a signature of selection. But to be more precise and more correct, you cannot simply use the diversity index to claim for a signature of selection, because you should use a different statistic, different parameters that allow you to prove that this reduction in diversity is significantly different. So now we just compare the diversity. We simply check for diversity. We are not going to compare the diversity. And to make a second step in this analysis, we ran a new mathematical experiment using 3 different indexes. And you find here some details. I'm not an expert on this kind of work, but in principle, each index works on a different approach. And we use 5 different indexes because we were looking for a signature of selection supported by more than one of those indexes. And again, I give you some example. Well, this is a comparison between wild MR wheat and durum and domesticated MR wheat. Each circle represents different indexes. So it's from diversity reduction index to this up FLK. And again, we can go more in details. For instance, here, this is the chromosome 6A again. And you find that in this region, that the same region I showed you before, in the chromosome 6A, at least 4 different indexes suggest the presence, give a significant result, and so suggest the presence of a signature of selection in this specific region. And if you look to the genome and you project this data on the genome of the durum wheat, you find that in this region there is a famous locus controlling a grain weight or seed size. And so that might be an indication that the first domestication has also implied a selection of a region that control, improve, obviously, the grain weight or the seed size of the domesticated plants. This is a clear, strong indication of a signature of selection. The signature of selection is very large in the region, but because we are close to the centromeric region, so it's very often the centromeric region have a low recombination frequency. And perhaps there are also other genes, not only this gene which is known. But so far, there's a clear indication there's a signature of selection which is associated to the comparison to the transition from wild MR to domesticated MR. Some other example. So we are comparing domesticated MR versus durum wheat landraces. And here you find another strong suite of selection, which is supported by at least 3 different statistics. And there is a number of Cattivelli map on this region for many different traits, so it's not easy to identify, but there is many different regions, this very large region with many genes inside. Or we can have another example here where we compare durum with landraces and durum with cultivars. And again, there are an example of a strong suite of selection, which is supported by at least 2 parameters in this region, where again, in this region, there are a number of genes which are probably involved. This SUS, for instance, is a gene controlling still seed size, actually, sucrose transporter. Okay, so overall, you can scan the entire genome using the data we produce, and region by region, you can look if there is a signature of selection during one of the transition from wild MR to modern varieties. And eventually you can project this information on the genome of durum wheat and see if there are any specific gene under this suite of selection. This is a long ongoing work, so we are actually working on this data as well after the publication of this first data set. And day by day, we found new genes, a new suite of selection that might explain the evolution. But the important point is that this atlas of data prove that the domestication of durum wheat has an impact on the genome, and the impact is different from chromosome to chromosome, and also is different and according to the specific comparison you make, so the different step which has been taken during evolution from wild MR to modern cultivars. Okay, so this is a very nice tool to understand the evolution of tetraploid wheat, so the durum wheat, the durum genes. But at the same time, this panel can be used for other approaches, for other types of experiments. And here I want to present you a data. It was actually part of the genome paperwork, and this came from the lab of Curtis Postiak in Canada. And this example is nice because it tells you how we can use this very well-organized population to trace back the origin of specific allele. And the example I'm going to give you is a gene controlling grain cadmium accumulation. So cadmium, of course, is a potential threat for human health. And what is peculiar of durum wheat, that durum wheat, and not for instance red wheat, very often durum wheat cultivars, when grown on cadmium-rich soil, accumulated cadmium in the seed. And this can be a problem for commercialization, for trading and human nutrition. And in the past, a major locus controlling cadmium accumulation was found on chromosome 5b, so the locus was already mapped. And then recently the gene was cloned, and the gene clone is called HMA3B1, and is a gene coding for a metal transporter. And this we use, and the 2 alleles of this gene, the mutated forms, the allele that determine high cadmium accumulation is due to a mutation of the wild allele, and the mutation is simply a duplication of a team-based pair that introduces stop codon in the protein. So this is a knockout mutation, basically, and the plants carrying this mutation, this knockout mutation, accumulate cadmium in the seed. So we use this information to trace the origin of this mutation in the panel of material we developed. And this is what we found. For instance, so if you start from wild MR wheat, so green means low cadmium, red means high cadmium. If you start from the wild MR wheat, all wild MR we sampled, all of them were at low cadmium. So we did not find any wild MR carrying the mutation that that produce high cadmium accumulation. Then, if you move from wild emmer to domesticated emmer, you find that some domesticated lines start to have some mutation. The most frequent subpopulation, frequent cluster, is this one here from in this region, where there are many domesticated emmer with accession carrying mutation, and all these materials comes from the fertile creation from the southeast of Turkey, for instance. So there might be the origin of this mutation. So the mutation clearly was originated in a domesticated emmer, so in the field of the first farmers. But then, if you move from domesticated emmer from the fertile creation to the land races, you find that in land races there is a very high frequency of this mutation, and particularly those land races indicated here under the C segment are all land races coming from North Africa, Morocco, Algeria. So they are very popular. So, and this is coming from this region at a very high frequency of the mutation leading to high cadmium accumulation. And because when the people start making breeding durum wheat about 100 years ago, they start using as a founder the land races from North Africa. It's not surprised that most of the modern cultivars contains this kind of mutation. And that's because this kind of mutation was present in the founder, and the founder were coming from most of the most important founders were coming from the North African durum wheat landrace population. So again, using this kind of panel, you can trace the origin of the mutation from domesticated emmer wheat to the modern wheat cultivars. It still open the question why this mutation has been selected. Well, there is an idea behind, at least an hypothesis. This kind of mutation allowed to the accumulation of cadmium in the seed, but also the accumulation of translocation of zinc in the seed. So the idea is that in zinc poor soil, this kind of mutation is probably an advantage, represents probably an advantage. And plants with this mutation probably grow better and at a higher yield. So for this region, probably the mutation was selected. And unconsciously, the people were also selecting for high cadmium accumulation in the seed. Okay, so this is clearly the first tool we developed. And this tool can be used, as I told you, for evolutionary study, for tracing back the alleles, and also for finding new alleles, if you like. But then I want to take a few minutes to present the other tool we developed, and this is called Global Durum Panel. This is a breeding dedicated tool, mainly focused on land races and modern varieties. The idea to develop this tool was really coming from a discussion in the expert working group for durum wheat in the frame of the wheat initiative. And behind this idea, there is a bottom-up approach. I mean, this panel is being collected, not controlling the biodiversity existing in the database. Rather, this was collected with a call from breeders. So every breeder contributed with its material. And the idea was to have a material representing what a breeder considers a most useful genetic diversity for breeding program. This is material represent the genetic diversity which is being used by breeders and pre-breeding in breeding and pre-breeding approach in most of the durum wheat breeding program worldwide. The panel has been genotyped, and now I'll tell you how we organize this panel. So the panel this was sponsored by Wheat Initiative actually, and the panel started in 2015 when we made a call for contribution, and about 2,400 sessions were sent by durum wheat breeders. Material was collected by Filippo Bassi in Carda, and we did the first nursery, the one you see here. And we checked for genotyping, seed increase, some preliminary phenotypic data. Okay, then we make a first genotyping using only 94 Cas marker, and we use this initial genotyping to make a selection to avoid, for instance, duplication, and to try to reduce the number for 2,400 to a smaller amount, and the final number was about 1,000 sessions. And so we try to use to make a genome-based representation of the material sent by breeders. This is the phyla composition of this panel. Again, we have some representative of dicocoides and tetraploid relatives, not so much. There is some durum-wheat landraces, much more. But then you see that most of the material represented by durum-wheat cultivars and coming from different breeding programs in the world. So we have cultivars durum-wheat from Canada, and we have one from French, Italian, and Central Europe. And then we have a lot of materials from Simit, Icarda, and other Mediterranean breeding program. So this really represents the diversity that the breeders are using currently in the breeding program. And this allows us to compare the different breeding programs and to see how breeding program differs in terms of genes and alleles. Okay, so again, we did this population structure analysis to see how the panel can be organized. And see clearly the first level of separation between emmer and durum-wheat. So emmer is the yellow segment, durum-wheat is the blue segment. But if you move forward, you see, the separation, you could start to separate the land races of durum-wheat from the modern cultivars. So the land races from durum-wheat is the orange segment, the modern cultivar is the blue segment here. And again, we can move forward. Okay, for the separation between durum-wheat, different breeding programs. So you have a separation between Icarda and Simit breeding program and the other breeding program. So again, the other breeding program are presented by the red segment here. Simit and Icarda breeding program are presented by the blue segment here. And you know, Icarda and Simit, they work together to some extent. So it's expected they share some part of their gene pool. Then you can move forward and you can have a separation between different breeding programs. So again, this analysis tells you that indeed our population not only represents the different breeding programs worldwide, but the different breeding program can be to some extent separated based on molecular markers data. And so this opens the possibility to compare the different breeding programs to see how breeding program differs one from each other at genetic level. Well, this is the neighbor joint tree analysis. Again, as expected, we found that there is from one side the emmer and the emmers in general, then we have the land races, and here on the other side there is the durum-wheat cultivars. This is pretty as expected. Again, this kind of data is in agreement with the previous analysis. And here you can see already, if you look to the legend, that you can try to separate the different breeding programs. So the durum-wheat cultivars, the cultivar durum-wheat, can be separated already in different subclusters, small clusters, representing different breeding programs worldwide. Again, this gives you the idea that the breeding program for durum-wheat in the world are not exactly the same, do not share exactly the same gene pool, but they have some specific genes confer adaptation to specific target environment. This is expected, but this kind of analysis really highlights very nicely this kind of organization. This global durum panel has been multiplied by Fibonacci and Icarda, and the seed deposit has been established in the Icarda gene bank. What is relevant for this kind of material, the global durum panel, is that the global durum panel is a publicly available resource. So you can actually ask Icarda to have a copy of this material, and actually already we distributed more than 40 copies of the material worldwide. There are many researchers worldwide are working on this panel, and each of them is trying to organize a phenotypic data for the traits of interest. And then if you do in this way, you have a phenotypic data, molecular markers, and phenotypic data are available and can share with everybody. And so everybody can use the common information in terms of markers and germplasm sources, add its own phenotypic traits, and then you can make the analysis very easily. So it is a way to involve many people to organize a sort of common general effort for improving durum-wheat breeding and genetic mapping. Okay, so what can be used for? Well, this kind of panel is clearly perfect for gene mapping and gene discovery. So you have a trait of interest, you want to find a resistance to a disease, or you want to find some of the diversity for your specific trait, you can use this panel, the molecular data are available, you run the phenotype experiment, and then you get the genetic analysis done. very simple. But I want to give you some example now how you can use this panel to study the relationship between different breeding programs. And for instance, you can use the standard statistic. I had one of the statistics before the diversity reduction index, and you can compare, for instance, the land races population versus culture population, and you find that there are some peak of this index which correspond to signature of selection, so diversity that has been selected, or reduction of diversity actually, that is found in the comparison of the two panels. And for instance, you can find well expected a key genes for a reduced plant height that is being selected in cultivars. There is a strong reduction compared to land races, or another gene here which controls is a sucrose transported that in turn increase the seed size. Again, it's well expected that people working on cultivars selected for larger seed and smaller plants. So again, this is a way to you can compare probably at the level of details higher compared to the one I showed you before in the previous panel. And you can identify genes as being organized, being preferentially selected in cultivar versus land races. But also you can do the same experiment comparing different cultivars or cultivars from different ages. So here in this very small comparison, we use we compare cultivar from the 70s versus cultivar of the 90s. And there are some peaks coming up, and probably the most curious one and most significant one is this one here at the end of the chromosome 7b. It is one of the gene F2N synthase controlling yellow pigment content, and it makes the semolina much yellow compared to the semolina of 20 or 40 years ago. This is a trait, is an industrial interest. So during the last probably 40, 50 years, the breeders start to select for yellow content, for yellow trait. And one of the first options was to fix a genes controlling for the synthesis of the pigment. But if you compare different breeding programs, for instance, breeding program from Central Asia versus breeding program from France, again, you find out the same gene as a peak. So clearly, the breeding program from France was most advanced in terms of selecting high pigment cultures compared to the breeding program from Central Asia, where probably the trait is not so relevant at the market level. Again, you can compare different breeding programs and get some information about the genes selecting a specific breeding program. Or you can compare overall the breeding program, and you find something that, like this kind of analysis that is probably expected. Again, it's not so surprising that there is so the relationship between breeding programs reflect a common pattern of environmental adaptation. So for instance, all breeding programs from Mediterranean region, Italy, Spain, Icarda, they share some similarity in terms of genetic diversity. And that is somehow different from the breeding program from Central Europe or North America and France, which is still a very northern country compared to Spain or Italy. And again, you find that breeding program from Spain, for instance, are very different. From Australia and similar, they are completely different from the other one. So there are some relation between breeding programs. And if you want to understand more, which is the genetic base of this relationship, you might be interested to understand if there are unique alleles in this breeding program. So we look for existence of unique alleles. So unique is an allele that occurs only in one of the in the germplasm of one of the breeding program and not in any other. So for instance, if you take the breeding program from Italy, we found about 100 unique alleles, or alleles that have been found only in the Italian germplasm. And out of these 100 unique alleles, only one allele had a minimal allele frequency higher than 5%. So most of these alleles are not only unique, but they're also very rare. They are higher than 5%. They are also very common. Sorry. So in Italy, there are 100 unique alleles, and those alleles on average are very popular, so they're not very rare. On the contrary, if you look to the same for Icarda breeding program, but if you look to the breeding program from Asia, Central Asia, there is a very high number of unique alleles, but those unique alleles are also very rare. So unique SNP showing in high frequency in a specific breeding program may represent breeding targets for adaptation. to the corresponding environmental conditions. So taking this kind of results, you can imagine to look more in details and to see what are the genes make the breeding program for a specific region different from the breeding program for another region. Okay, so this is I think I give you an example what can be done from this kind of panel. So our idea 5 years ago when we started this work was to develop really tools to make the analysis of genetic diversity in tetraploid wheat in a collaborative way and focus it on the major target, scientific target we can have, like evolution and breeding. And those two tools together really provide I think a comprehensive germplasm platform to support durum wheat breeding. And we think is an important tool for also for collaborative work. And we hope that this work will go forward in the next future. Here I've acknowledged a lot of people. So those are the people who were involved in the first paper dedicated to the first panel, so the global tetraploid wheat panel. And those are people, most of them were part of this paper on dedicated to durum wheat genome. And then recently we did this other paper on the global durum wheat panel. This is the breeding panel for the panel for breeding, and this is made by still a large international collaboration. And this was really made in the frame of the Wheat Initiative and the Expert Working Group for durum wheat. That's all for the moment. I thank you for your attention, for this great opportunity to talk with all of you, at least virtually. And thank you for your time.
Thank you, Dr. Luigi, for this great talk and which is very rich. I personally learned a lot about this panel and about this initiative that seems very interesting, not only for the present but for the future of the breeding of wheat and tetraploid species of wheat. So for the question, the first question is by Dr. Muhaf Rahi, my colleague. It's related to the magnitude of the genetic diversity reduction in B genome as compared to A genome. As you know, the B genome is rich in heterochromatin, which is generally gene-free region in the chromosome. This may lead to less reduction in genetic diversity. Can you confirm this or not?
Well, we did not find a clear trends in between the two genomes. So I agree, the B genome is very complicated, very difficult. Also, in terms of sequencing, the B genome is more fragmented. So that is maybe a bias in this kind of analysis, because the integrity of the genome is really helpful in this work. But in principle, yeah, there might be specific region, but it's not, there is no clear trend of the two genomes. What you find that there is a many very large region, this kind of genetic diversity. Most of these large regions are organized around the centromeric region because the recombination is very low. But I cannot say that there is a trend where a B genome and the reduction is different compared to the A genome.
Okay. I have a question because for me, personally, I've been working on livestock genomes, which for sure there are many differences between plants and animal species at the genomic level. I saw that the genomic regions that are under selection by different metrics that you were using, I think the genomic regions are very large.
Yeah.
Is there any explanation for this?
Yeah. Because the durum wheat genome, the wheat genome in general, is made of very large chromosome, and so a chromosome may be 0.5, 0.8 megabase. So the entire genome of durum wheat is 11 gigabases. Sorry.
Gigabase. Okay.
So a single chromosome spans about 0.7, 0.8 gigabases. So it's extremely big. And those kind of chromosomes are very rich of heterochromatic region in general, and the recombination is very is distributed, is not uniformly distributed. And the centromeric region, that is very large, has a very low recombination frequency. I mean, probably in the centromeric region, the region of low recombination frequency is something like 1/3 of the entire chromosome.
Mm-hmm.
Well, this is a huge. So if you have a gene which is being selected in the centromeric region, under selection in the centromeric region, the entire centromeric region means about 1/3 of the chromosome is fixed. And this is exactly what we found in our analysis. So very often we have large region corresponding to the centromeric region which has been fixed during selection. Probably because there's a single trait or few traits of relevance in this region, but the entire region has been fixed because the recombination frequency in this region is almost absent, very low, extremely low. That is a very typical features of wheat, because also barley. So this kind of crop with very large genome and where the recombination is completely focused or concentrated in the telomeric region of the chromosome.
Yes, each Aikin effect is more more strong.
To clone a gene in the centromeric region is a nightmare. Okay.
Yes. Okay. I have another question about when you mentioned about the unique alleles in different breeding programs. I saw a great difference between the nature of this, the kind of this rare of these unique alleles in Central Asia and in Italy, for instance, and what is surprising is the percentage of rare alleles in the two breeding programs. Is there any explanation? What happened to get this result?
If you have many rare alleles, it can be simply an indication of the diversity you have in this population. We actually were interested particularly in a unique allele not rare, because a unique allele not rare might suggest that there is relevant for the adaptation of the target environment where the breeding program works. We found also some unique allele covering about 50% of the session. I mean, that means that this unique allele in some breeding programs really represent an indication, or might represent actually, an indication of an allele which is relevant for the adaptation to your environment or for specific traits being selected, particularly in one breeding program. But I think we can interpret the presence of many unique allele, a low frequency, unique rare allele, as an indication of residual diversity that you have in your breeding program.
That is, which is threatened.
Yeah.
To be lost.
Yeah, of course.
And never recovered. Okay.
Yeah, this is also can also be a problem.
Yes, okay. The following question by Dr. Farahi: is there any selection in pH gene in durum wheat?
You mean pH is pairing homologous, is the gene which allow the pairing of homologous gene, the homologous gene during meiosis? Well, no, there is no indication for any selection. I mean, all cultivated, all material is pH, so it is as pairing as the pH gene. There is no indication of specific selection. Yeah. Also, wild material has the pH gene. It's not something which should be introduced during domestication. So all wild them carry the standard, the wild type pH gene.
Mm-hmm. I have another question about the cadmium accumulations. What I was thinking, I know that this metal is not good for health, but you said that it's good, and the results about the North African cultivars, it was surprising. I think maybe I didn't follow yet. Good.
Okay. Well, cadmium is toxic, so no doubt. Cadmium is toxic. This mutation increased the accumulation of cadmium in the grain, so potentially is a negative mutation. But what we find out, this mutation was originated in a domesticated emmer, so already in a plant that was grown by farmers, and became very common, so was basically selected unconsciously by the farmers in the past, particularly in some region of the world. The idea, because the possible explanation of this very unexpected finding is that the farmers did not select the plant because of the high cadmium in the seed, but because those plants, at least the transporter, the cadmium transporter, not only transport cadmium, but also transport zinc. And so if you have a soil which is poor in zinc, to have an increased accumulation of zinc in the upper part of the plant is an advantage for the plant. So there are some experiments showing that if you grow plants with the mutation, so plants carrying accumulating cadmium in the grain, in a soil with very poor zinc, those plants perform better than a corresponding isogenic plant. So it means the selection was probably carried out because of the higher performance in terms of zinc accumulation. Of course, nobody was known about the cadmium at the time and was selected. And clearly the cadmium phenotype is evident only if you grow the plants in cadmium-rich soil. If your soil is poor of cadmium, there's no problem. And this problem is particularly important in some regions, like in Canada, for instance, where they have a soil which are rich in cadmium. They don't have problem in terms of zinc, but because they have cadmium, if they grow this kind of plants, they accumulate cadmium in the grain. By experience, I can tell you in Italy, there are very few cadmium-rich soil. So for us, even if the plant is potentially capable to accumulate cadmium, there is no problem because there is no cadmium in the soil, so no accumulation happens. But the original of this selection probably was not because of cadmium, but because of the concomitant accumulation of zinc, which is an essential component, it's an essential mineral for plant growth. Okay,
that's clear. I have another question, which is methodological indeed. Why did you use these three metrics to detect selection signatures? XPCLR, XPHH, and APFLK, and also FST. Okay.
When we submit the paper at the origin, we use only a couple of metrics, so the FST and the DRI. Then the reviewer said us, well, you should prove the same result using different metrics. So we put together 5 different metrics, and the idea is to trust only regions which are confirmed by at least 2 different metrics. Indeed, it's very difficult because the metric works on different principles. So it's very difficult that all 5 metrics give the same results in the same positive region. Indeed, I think we don't have any example, but we have example of region indicated by 3 or 4 metrics. So we trust those regions for selective sweep, only the regions which are supported by at least 2 different metrics in the analysis. This is a way to improve the trustability of the data.
Mm-hmm. And you kept only the regions that were detected by at least 2 methods?
Yeah.
Methods, okay. Okay, good. What about the future of this panel, of this initiative? What is the impact on the durum wheat agriculture in general?
The second panel, this global durum panel, as I told you, has been distributed already to about 40 different scientists worldwide, from US to Asia. And the idea is to provide the durum breeders or the durum geneticists with a tool where they can simply test the phenotype, for instance, can be adaptation to your environment or disease. And then as soon as you have collected the phenotypic data, you can straightforward go to the marker analysis and the genetic mapping of the traits, because all the molecular data are freely available. They can be downloaded from Grain Genes, for instance. And so everybody tried to work on the same panel. In this way, our original idea was that all breeders can share knowledge, because if breeders in another side of the world identify a gene in a specific variety which is present in this panel, at the same time, breeders on the other side in another country have already the gene in his field, and using the marker can take the variety they are already and start breeding. So this should be a sort of common tool to deal with genetics in durum wheat. So this is the original idea. I hope we will be able to achieve something. So far, there is a network of about 20 field trials around the world, and try to organize some common phenotypic data, and they are working to produce some results. So far, we published only the panel and the comparison of different breeding program. But the panel now is open for phenotyping, and we also are running some phenotyping, but not only our group. There are many groups also in your country, for instance, but in North African country, in Argentina, US, Canada, and also in Asian country, using this panel to map traits.
Okay. So do you recommend to use at the end, knowing these results, do you recommend to use improved varieties everywhere, or should be used in a way that keep diversity and keep land races and so on?
Well, first of all, I'm a breeder, so I support breeding. It's not necessary that the modern variety has no diversity. I mean, modern breeding also produces diversity. Okay, we collect all this diversity, and there are plenty of gene banks worldwide where we keep and we analyze this diversity, and this is certainly true. And what I think is that we need to develop genotype according to the climatic condition of the target environment. I mean, very often I say if there is a climate change and tomorrow is warmer than today, there is no logic, there is no reason why a plant selected yesterday when the climate was cooler is the better plant for tomorrow when the climate will be warmer. So if you really believe in the climate change, you need to select variety for the new climatic condition. And this is clearly what also the message of this kind of work. So breeding programs from different regions of the world, they are not identical. So they've been tailored for the specific target environment. So if you take a breeding program from South Italy or Mediterranean region, is different from the breeding program for Canada. That's because the environment is different. And so if the environment changes, you need to change the variety. I don't say that you need to throw away genetic diversity. So we need to keep, conserve, and study genetic diversity, but breeding for new cultivar, in my understanding, is mandatory for the future.
Okay, thank you, Dr. Luigi. There is no other question in the chat. I thank you very much for this rich answers also. They were rich as your talk also. So at the end of this webinar, I would like to mention that the next webinar is scheduled in 2 weeks in the same time. My colleague Butaina is going to share the flyer of the next one. Okay, it will be entitled, it will be in French actually. In English, the title is Assessment of Technical and Scientific Cooperation in the Field of Agricultural Research. It will be presented by my colleague, Dr. Fatima Hammi, administrateur at INRA, Morocco. Otherwise, at the end of this webinar, I would like to mention that you will get a very short, actually 3 questions survey to get your assessment of this webinar series. Please fill it. It will be very helpful for us. Finally, on behalf of the organizers, I would like to thank everyone who participated today and thank warmly our guest speaker, Dr. Luigi Cattivelli, for his rich talk and rich debate. I would like to thank also my colleagues Butaina, Abidou, Atman Spatar, Tirazi, and Abdelaziz Yasri for their crucial contribution for organizing this webinar series. Thank you very much, Dr. Luigi. Would you like to say something?
Well, I have to thank you for this opportunity, and of course, I really hope to see all of you personally, not virtually, but I look forward to have the opportunity to meet you and your teams in the near future.
Yes, looking forward to hosting you in another webinar. Thank you very much.
Perhaps in a real seminar, not a webinar.
Yes, real seminar. Okay, perfect. So see you later. See you in 2 weeks for the audience. Bye-bye.